Mitosis is the process by which somatic cells divide. It is essential for the maintenance of life. Mitosis is responsible for growth in an organism. The genetic material of the nucleus is duplicated so that the daughter cells produced are genetically similar. The cell cycle is composed of 4 parts, the G1, S, G2, and M phases. Interphase is the combination of the G1, S, and G2 phases. The cell spends the majority of its time in interphase. In G1, the cell grows by replicating organelles and cell material. In the S phase, DNA is synthesized and the chromosome number doubles. During G2, the cell continues to grow by replicating organelles in preparation for the M phase. M phase is the meiotic phase where mitosis and cytokinesis occur. Cell division is the purpose of the M phase. Mitosis is made up of prophase, metaphase, anaphase, and telophase. This is followed by cytokinesis. In an animal cell, during the beginning of the cell cycle in interphase, the cell has centrosomes with centriole pairs, a nucleolus, nuclear envelope, and chromatid. In prophase, the chromatin condenses into identical sister chromatids or chromosomes. The nuclear envelope begins to fragment and the spindle fibers form. Kinetochore and non-kinetochore microtubules also form. The centrosomes move to opposite poles by the aid of microtubules lengthening the cell. In metaphase, the centrosomes have now moved to opposite poles. The kinetochore microtubules attach to kinetochores on the chromosomes, and the spindle fibers attach as well. The chromosomes line up across the metaphase plate. During anaphase, sister chromatids are pulled apart to opposite poles. The kinetochore microtubules shorten while the non-kinetochore microtubules lengthen the cell. In telophase, daughter nuclei form on each side of the cell and the nuclear envelope begins to reform. The chromosomes begin to unravel and the cleavage furrow appears. In cytokinesis, the cytoplasm is divided. The cleavage furrow deepens and the cells are split in two. 2 identical daughter cells are produced. The daughter cells have the same number of chromosomes as the first cell. In animal cells, the cleavage furrow allows for cytokinesis.
Meiosis is the process of cell division in sexually reproducing organisms that reduces the number of chromosomes in reproductive cells from diploid to haploid, leading to the production of gametes. Before a cell can enter cell division, it needs to prepare itself by replicating its genetic information and all of its organelles. All of the preparations are done during the interphase. Interphase proceeds in 3 stages, G1, S, and G2. Cell division operates in a cycle. Therefore, interphase is preceded by the previous cycle of meiosis and cytokinesis. In prophase 1, the 2 chromosomes become visible and they combine to form tetrads. The 2 chromosomes cross over at points called chiasmata. This allows the chromosomes to exchange genetic material. By metaphase 1, the nuclear membrane has disappeared and the microtubules form a spindle. Spindle fibers attach to only one side of each centromere, and the 2 homologous chromosomes attach to microtubules orienting from opposite poles. In anaphase 1, the microtubules of the spindle fiber shorten and pull the chromosomes towards the poles, taking both chromatids with them. Each pole ends up with a haploid set of chromosomes consisting of one member of the homologous pair. During telophase 1, the nuclear membrane reforms around the daughter nuclei. Each daughter nucleus contains 2 sister chromatids for each chromosome. Because of crossing over, the 2 sister chromatids are not identical. In cytokinesis, the division of the cytoplasm and the plasma membrane following the division of the nucleus results in 2 cells, each having its own nucleus and cytoplasm surrounded by a plasma membrane. The purpose of meiosis II is the separation of sister chromatids and to create 4 genetically diverse haploid cells. In prophase 2, the nuclear envelope is again dissolved and the spindle is set up again. In metaphase 2, the chromosomes move into the center and line up. Now there are 2 chromosomes instead of 2 tetrads, and the chromatids will separate. In anaphase 1, the microtubules of the spindle fiber shorten and pull the chromosomes toward the poles, taking both chromatids with them. Each pole ends up with a haploid set of chromosomes consisting of one member of the homologous pair. In telophase 2, the chromatids concentrate in the poles and the nuclear envelope is reformed and the spindle again is dissolved. The cells divide for the last time, leaving a total of 4 haploid cells, which have half the chromosomes of the diploid cells. Unlike the daughter cells from mitosis, the daughter cells produced here cannot immediately cycle back to interphase.